Thank you so much. We'll hold questions for the panel. And now I'd like to introduce our last speaker of session one, Dr. Luoping Zhang from University of California at Berkeley. She will be speaking on applying CRISPR in environmental health research from cells to human populations. Thank you.
So I really like to thank the National Academy to organize this very timely and exciting workshop. So this morning, the previous speakers already talked about the basic biology of the genomic editing. And Rick just from NIEHS talked about challenges and the promise. And I feel I may not need to even give any more talk. So they've, prove all the foundations. So make my talk is much easier. So I'm just going to try to talk about how is our approach to apply CRISPR system in the environmental health research. So but today I'm going to just focus on the approach and not really data. And my collaborators and this afternoon would give more details. How can we use this? Where to? Okay.
That's for pointing.
Okay.
Then, yeah.
So there are three major genomic editing tools, and I don't have to go through all the zinc fingers and talons. So today I'm going to only focus on the CRISPR. How did we apply for it? So CRISPR has applied to many, many different areas, especially lately, and including gene silencing amplification, and also we know that develop the new cancer treatment. For example, we already know there are clinical trials for lung cancer treatment with CRISPR in China and in the US, and also recently, CRISPR, using CRISPR screen already identify 100 essential genes for the cancer immunotherapy, right? So, and also what applied in, so this is a very good base to apply to the precision medicine and really, close to our environment, like, destroy the virus like a Zika virus and HIV, etc. And also, CRISPR was involved and applied in the agriculture, trying to make more, improve the food and security. And now, I like to just say a few words on how the CRISPR also used in the trying to improve or make the new, more clean biofuel. So recent studies, they showed using CRISPR gene editing in yeast strain or in agar that can produce a new precursors and more conversion from sugars to lipids. So that's the basis for the sustainable biofuel products. So I'm just going to point that out. That's better for the environment. But today my talk is really trying to focus on toxicity testing for the environmental chemical exposures. So that's my talk. So I borrowed my old slides 10 years ago, and here in National Academy, and hosted the workshop for 21st century toxicity testing. So proposed that, trying to use a more efficient in vitro testing system and based using array-based toxicogenomics approach. That's 10 years ago. 10 years now, but that time, 2007, CRISPR has really not very popular. So we can look at this data I'm showing. Just take a quick PubMed search last week. You see 2007, the CRISPR application is still very low. But 10 years now, you can see the CRISPR paper published is really exponentially increased. But if we look CRISPR and the environmental health sciences, and the paper is still very, very limited, especially if we're trying to testing chemical in the environment, exposure is very limited. So I'm just giving you a couple examples for published very recent years. There is a group in China, that's what I found the very first testing the triglyceride in the human cells by using the CRISPR screening. So very recently, there's another group in Chicago also using a CRISPR screening to testing the paraquat induced for the cell death. So look at, so this is approach, try to using CRISPR technology to screen toxic chemicals and try to identify the response genes and identify the pathways, the chemical exposure involved. So this is a approach is trying to see what's the susceptible genes and why we needed to screen for that. So we know for the maybe not even 2 decades, we were trying to identify susceptible populations who are sensitive to toxic chemical exposure. So we see from the different populations, they carry the different genetic variations, they would make them quite different. So for example, the chemical exposure for more susceptible people, you may develop a disease. But for less susceptible, then you have no disease. But how could we identify this population? Well, for the last 2 decades, what we really use is called the GWAS, right? It's trying to using the GWAS strictly apply to the population, what's diseased people compare with non-diseased people, and then they see what genes. But this approach has advantage to discover some new non-hypothesis, some mechanism or some genes we don't know to involved with this disease. But it's not efficient, which means the GWAS needs to apply to a huge population. And then it's a lot of money involved, and it's taking long time to really try to identify a susceptible population. So here at Berkeley, and with my long-term collaborator, Dr. Chris Luoping, and who is going to give a talk this afternoon, give more details about our study. So here, we're just trying to focus on our approach at UC Berkeley. It's funded by, of course, NIHS, the Superfund Research Program. So we both correlated this project to functional profiling of susceptibility genes. So what we were trying to is identify the susceptible genes, not from human directly, but from the human cells. Then apply what we have learned from human cells to the human population. So that's what our approach. And then on the other hand, what we were really trying to do is putting the genome editing into the toxicogenomics. 10 years ago, when the 21st century toxicity testing, all array-based, I think that time mostly is focused on the microarray, right? So microarray. So, but our approach is actually trying to using the functional genomic screening, first using CRISPR's yeast system, okay? Screen genomic response in the yeast system, then we trying to conform in the human cells by RNAi, then we moved from the yeast directly to the human cell using this specific haploid human cell line KBM7. So, and then most of the currently, we were applying CRISPR in the human cells. And so this is the work we were really trying to summarize and in this review paper. And then that time we were really trying to advocate the importance of application of the CRISPR using in this environmental health sciences. So that's our new approach, how our approach works. Again, so this is approach testing by CRISPR and from so collaborating from University of Florida and UC Berkeley. I'm just going to quickly show using arsenic as an example. So what we were trying to do is propose 3 steps. The first is a genomic-wide screening. You can screen using CRISPR-I and A, or also you can use a more targeted approach. So that's a screening, step 1. Step 2 is after screening, how do we really know what we have found or the genes are real, truly response genes. So we have to do, different layers of validation. Then third is how we can apply what we learned from the cell system to the human population studies. So the genomic-wide screening, we propose kind of 2 approach. One is the genomic-wide CRISPR screening. So at Berkeley and UCSF, the IGI, they developed this innovative genomic institute, developed this 2 genomic-wide screening. One is called CRISPR interference. It's all, the pool of the cells is a loss of the function for each gene pools, or CRISPR-A, the activation, the gain of the function. So then the goal is trying to identify responsive candidate gene. So this is, so I'm show you another principle, how that works. And Dr. Kapman from UCSF recently published quite a few papers on how they apply both CRISPR-A and I in their study, particularly in the neurodegenerative disease. So here is basically how can I go back? Okay. Yeah. So basically, this approach can tell, you can see using CRISPR is very flexible. You can try to lock out the genes. You can also try to amplify the genes. And you can mix using, create the library, sgRNA library, with either genomic-wide or targeted. And in the cells, any cells you're interested. So that's the flexibility of CRISPR. And also, after you generate the good cells, you can treat your cells with the chemicals of your interest, and then you screening and sequencing, then you can identify what genes has the most responsive to the phenotype you're testing. So this is basically the approach. So for now, what we have seen, this is a genomic scale, the CRISPR-I or CRISPR-A screens is available in these human cells, and also with quite different phenotypes. And mostly is for the cell fitness, cell growth or cell death. So this is just to show you what's available right now. And also at Berkeley, the IGI, they also provide the targeted, so or we can say the sub-library of 7 different sub-library. So here, and it's a pair. So they each sets, they have either interference or activation. And if you are interested for the more information, you can contact Mary West. So I'm just going to show you what's available there. So IGI and also at Berkeley, they provide this library for all the researchers to do the research, to do the work. And now we're using what's a targeted approach for the CRISPR? Like what we have heard, many of them you can using, specific one single gene targeted. For example, for now, we are testing for meta-high toxicity by the locking out the FUNC-D2 genes. And also you can target on the group of the genes of your interest. For example, I just introduced the RGIs, the 7 subsets of the library. But really most important, I think, is in collaborator with Dr. Lu and Dr. Luoping, Dr. Lu from Harvard, Dr. Luoping from University of Florida. We generated this as a specific group, specific we call CRISPR. So this targeted approach was actually focused on more than 3,600 genes, and the genes come, based on the NIHS and the NTP Tox21, the top 1,500 plus genes. And so that's including all that genes selected from the Tox21, and also plus some genes from environmental genome projects. And also we add the genes often related with the toxic chemical exposure, and we know this gene was a response to exposure-related disease. So that's we put this together. So, and what we also want to point it out, this targeted approach, we can use it either for screening. If you have more targeted hypothesis, you don't have to do genomic-wide scanning. So, but also we can use this targeted approach for the validation as well. So how to validate the candidate gene in the human cells, we think is important because especially for genomic-wide, you want to really make sure the genes you selected is not by chance. And so there is a few different ways you could do it. Of course, you can repeat the genomic-wide screening, but we don't think that's a very economic way to do it. But what we're trying to is when we make it, for each gene, we have multiple probes and target on the different site. So you can looking for if the same gene has a different multiple probes, they all can respond to your testing. So that's a good sign. And also we can using the Tox CRISPR. So also after this, we're trying to collaborate with our biostatistician and the bioinformatics, and trying to really using a more efficient way to select the real gene. Then we're trying to, from the gene, we are trying to make the pathway analysis and trying to discover what's the real mechanism of action. So the question now is how can we validate the functional and the mechanistic rules. So we're using 2 approaches. One, in vitro testing and in vivo testing. So that's basically we're trying to do the more biological function testing. So that's basic promises and the challenges. And since Rick already talked about, and here is what I have to try to put a few of my personal things here. For the challenges, of course, many people already talked about the off-target effects. So that's still a major thing, even though the technology has been improved. But also, I think the data quality control is another importance. And since the NIEHS was a sponsor for this, I want to point out the limited funding. And because we haven't get to the human study because we couldn't even get the funding. So we were still in the cell screening system. So without delay, this is our summary, how to, from cells to the humans. And this is just the summary from the genomic-wide screening and the validation from the targeted approach and biostatistical and pathway analysis. And finally, we could apply what we have learned from cells and then tested in the human studies. And again, it's a collaboration between Berkeley and Florida. Thank you for your attention. And of course, Chris and Jeppercorn is from the IGI as big supporting from our study, and Dr. Chuan Lu from Harvard helped us to create the CRISPR library. And and sorry, it didn't come up. Okay. The NIEHS is our funding source. Thank you so much.